Good day, students. Today's lecture is going to be on page 87. It had to do with turbidity measurement of broth cultures. And in this technique, we're going to use the spectrophotometer. Now, the spectrophotometer is playing a very important role in microbiology. Most of the time, we're using it to measure a bacterial population. Is a bacterial population low? Is a bacterial population high? Now, how is the spectrophotometer working? It has a globe, and then it passes light through to a photocell. So the photocell absorbs the light, and then it reads the absorbance for us. Actually, it reads the optical density, but we can convert optical density into absorbance. That we will see on the next page. On the next page, they show us the formula to calculate optical density of the percentage transmission. Okay, now optical density and absorbance is exactly the same thing. Percentage transmission. Some spectrophotometers read for us percentage transmission. Okay, so if the spectrophotometer is reading percentage transmission for us, there's the formula to calculate optical density, and optical density is obviously absorbance. Now, what is happening? This photocell and the globe, between it, we're putting what we call a cuvette. Now let me spell a cuvette to you. C-U-V-E-T-T-E, cuvette. Now this cuvette is normally from plastic or it's from glass. So light can pass through to the photocell. The moment we put a test tube or liquid into that cuvette, and there's bacteria inside there, the bacteria will bounce the light waves away from the photocell. So if it bounces now the light away from the photocells, so the absorbance is increasing. And if the absorbance increase, it means it is going to be a higher concentration of bacterial cells. So when we're doing growth of an organism in a liquid nutrient broth in a flask, on day 1 we will find that the absorbance is quite low. On day 2 the absorbance is increasing. Day 3 the absorbance is increasing more. And we can use this also a lot in the industry, especially where they're working with fermentation tanks and they want to know how many bacteria is present. Now, as I said in previous videos, that not one technique is perfect. There is techniques that gives us, all the techniques is just giving us a rough indication of how many bacteria is inside the cell. Why do I say this? You know, in this case of the spectrophotometer, dead cells will also bounce off light or scatter light. Debris inside that broth will also scatter light. So the actual reading is not always 100% correct. So what do I say? When we're doing pasteurization or sterilization with this bacteria inside, the absorbance will be exactly the same because dead bacterial cells, debris, will always scatter light. I just want to go through now quickly on this overview on page 87. On page 87, they say, it is possible to follow the growth of a microbial culture by measuring the number of parameters. For example, growth can be followed by viable plate counts. I will come in the next video onto a viable plate counts. And other ways include direct microscopic counts that we've done already, or we can do turbidity measurements. And that's what we're doing today, is doing turbidity measurements. turbidity is the effect of light scattering by a colloidal suspension. Many microbial suspensions are colloidal suspensions because the cells do not settle out of suspension quickly. These suspended cells scatter light, producing then turbidity. And this turbidity, you can see it with the naked eye also. As microbial cultures grow, the number of cells increases, accompanied by proportional increasing in turbidity. Now, microbiologists follow the change in turbidity with an instrument being called a spectrophotometer, now, which is measuring then the light that passes through a liquid medium. Now, that's another disadvantage of this technique, students, is that we cannot use solids. We can only measure liquids in this technique. The amount of transmitted light decreases as the cell population and hence the turbidity increases. Now, how this spectrophotometer is working is quite nicely described for you in figure 20.1 and how it's operating. But I want to stand still on the second paragraph on page 88 where they say, however, turbidity changes as a result of cell reproduction are not usually measured as transmittance values. Instead, measurement in optical density or absorbance are made. This is because optical density is directly proportional to cell or biomass concentrations. As cells increasing in number, so does the optical density. Recall that bacterial cells multiplying by binary fission, one cell becomes two cells becomes 4 becomes 8 cells becomes 16, and so it carries on. Optical density is also a logarithmic value, as may be seen by the relationship to transmittance. Now, if we, in certain spectrophotometers, if they're giving us a percentage transmission, we can calculate OD. There it is. OD equals 2 minus log percentage transmission. So if we know what is the percentage transmission, we can work out optical density, and optical density is the same as absorbance. Now, optical density is referred to as absorbance, okay, because spectrophotometers are often used to measure the concentration of color-absorbing solids and solutions. Absorbance values are exactly the same as optical densities. Yeah, biochemistry also use a lot the spectrophotometer. They're reading the concentration of glucose. They're preparing standard graphs for fructose and sucrose and so on. So, biochemistry also using it a lot. Now, what is very important, and take note of it, and that's what I always ask the students. I want to know, how can you as a microbiologist use a spectrophotometer? How the spectrophotometer is going to work, that must be general knowledge. I want to know, how can you as a microbiologist, how can you use this spectrophotometer to your advantage? And that is in that second last paragraph where they say, turbidimetric measurements to follow the growth of a microbial culture are convenient and rapid compared to viable plate counts. However, if growth results are required to be expressed as colony-forming units per volume of medium, this can still be done by use of a standard curve. That is so important. And they say, with values on the y-axis versus the number of cells on the x-axis. You can do it the other way around also. It doesn't matter. As long as it's a standard curve of absorbance versus the amount of bacteria. That is what it's all about. Okay, or you can put the amount of bacteria on the x-axis and absorbance on the y-axis. It doesn't matter. All right, but now, what can we do? And let me show you. I've done one now here. And on the x-axis, I've got absorbance, and on the y-axis, I've got bacteria per mole. Okay, now there is my figure. There is my figure. Now, you can see now, this is a standard curve, and let's say I'm going now to the spectrophotometer, and I read 0.3, I go up onto the graph, I go into the side, and then I see, yeah, it is 10 to the power 4, 10 to the power 4 bacteria per mole. Okay, and if it's 0.1, I can see roughly it is only 10 to the power 1 bacteria per mole. Okay, so we can use. Now, how do I get to this graph? The first time that we do this graph, every time that we're taking absorbance reading, we must do a plate count. And in the plate count, we find how many bacterial cells are there. And then we can plot now the plate count versus the absorbance. So it has got a very nice practical application for us. I just want to go further with you where they say, as with every technique, the tubertic procedure has also its limitations, and I mentioned already a couple of them for you there. And they cannot distinguish between dead or alive cells. Its sensitivity is restricted to bacterial suspensions of 10 to the power 7 bacteria per mole. So, for low concentrations of bacteria, this technique is not working because there's too little bacteria inside that suspension that can scatter the light. Okay, so there is a couple of other disadvantages, and on page 89, there is quite a nice picture of what is the spectrophoto look like. It is roughly about 30 centimeters by 30 centimeters, about 15 to 20 centimeters high. And we can then, it will show you a wavelength knob, a meter reading where we can read either percentage transmission or we can read the optical density. It's got a pilot light, a sample holder where we put the cuvette in, and a power switch and a light control knob, and then we can adjust the wavelength. Now normally we're working at a wavelength of roughly between 450 nanometers. Let me just see if they've got any other. Yeah, here in the book on page 89, they say they're reading the turbidity, they're reading at 620. At 620. Now, obviously, we must have a blank. The blank goes in first, and we zero the spectrophotometer. And when it's 0, we take out the blank, and then we start reading all the different tubes that we need. Okay, students, I hope that you can understand that the spectrophotometer plays a very important role. I will say after the autoclave and the microscope, the third most important instrument in a microbiology lab is the spectrophotometer. I hope you understand this, go through it, and make sure that you understand. I want you to keep safe, keep your family safe, keep everyone around you safe. listen to what our president is saying, and let's all stay healthy and safe. All of the best. Bye-bye.